I'm Daniel Levine, and this is the Bio Report. Researchers at the University of California, Berkeley and the Gladstone Institutes have grown beating cardiac tissue from stem cells in work that may lead to new ways to quickly screen for drugs likely to cause birth defects in the heart and identify drugs that may be dangerous to use during pregnancy. We spoke to Bruce Conklin, a senior investigator at the Gladstone Institute of Cardiovascular Disease, about the work, which was published in the journal Nature Communications, the promise it has for providing more accurate insights than animal models, and whether the approach could be expanded to other cell types to screen for drug toxicity to other organs. Bruce, thanks for joining us.
Oh, thank you for taking your time to be with me.
We're going to talk about the work you did that was published in Nature Communications to coax stem cells into cardiac tissue as a model of a developing heart. This has some implications for drug development, and I wanted to start there. We hear a lot about the high cost of drug development. Drugs fail in the clinic because they're shown either to be ineffective or unsafe. What role does toxicity play in the high cost of drug development today, and how much of that is the result of getting the answer later in the process after much investment has been made?
Well, I think that I'm not in the pharmaceutical industry directly myself, but I think that that they would tell you that it's the high cost of development, and the large part of the development is the number of drugs that fail. So if you could improve the success rate, then presumably the cost of development would be lower in terms of to find a successful drug. I would like to say that the pharmaceutical companies would pass on those savings, but there's also other market forces that are at play. But I certainly would say that that is one of the causes of the high cost. So I think that one of the real problems that we've had with testing drugs in the past is that we've relied on animal models for most of the toxicity. And although we're very similar to a mouse or other species which are used for these toxicity testing, that in fact that we're quite different when it comes down to the details. And particularly a mouse has a heart which beats at 600 beats per minute, and ours we'd be dead basically if we were beating at 600 beats per minute, and really the normal resting rate would be more like 60 or 70 beats per minute. So that's a huge difference in terms of the physiology of a mouse heart compared to a human heart. And so because the entire the setup of the heart is so different, it's not surprising that at a physiologic level they're very different, and therefore there are different toxicities and different susceptibilities to drugs. So the great advance which has occurred is that we can now use. First, we used embryonic stem cells, human embryonic stem cells, and now we use the induced pluripotent stem cells as the starting material. Those cells proliferate so we can get large amounts of them, and then we can make those directly into heart cells, and they're now human heart cells.
Why don't you walk us through exactly what you did?
So, the whole process begins when we take a normal human, and they have some. We can take some skin cells, any types of cells that would be used, the types of cells that would actually normally heal a wound. We just take some of those cells and they grow in a plate, and we can transform those cells into induced pluripotent cells, or iPS cells is what we call them. And the iPS cells are the ones which actually behave exactly like a human embryonic stem cell, but it's not from a fertilized egg. It's actually from a person who we know and we can examine and we can say is normal with respect to their heart exam and their so they have a normal heart, they have a normal they have normal mentation, all sorts of other things are normal about them, and that's something we don't know about embryonic stem cells, because those embryonic stem cells come from a fertilized egg, but we have no idea if it would actually grow a normal heart if we were allowed to. So that's a huge difference, is that we're starting material is actually we absolutely know would make a normal heart, and that or other body parts. And then what we do is initially it was very difficult to make iPS cells or embryonic stem cells into cardiac cells efficiently. So when we started off in the mid 2005 with this sort of work, we have a good day would be 1 in 1,000 cells would be a little beating heart cell in a corner of a dish, and we could actually study those cells, but it was very, very difficult to actually do experiments because there were just a few cells and they were in a sea of other types of cells. Over the last few years, we've been able to develop techniques so that we can make now beating sheets of cells, and this is from work from people all over the world that have been working on making this method better, and now we can get a sheet of cells that are beating at the bottom of a plate, which are now almost 100% cardiomyocytes, heart cells. And so that was the beginning part of when we began to start with collaborate with Kevin Healy. But
how do you get these cells to differentiate and organize?
So we have the way those initial cocktails were developed is it's sort of like cooking. People try different things and then the cells become changed. We know through using developmental biology what some of the key signals are for turning a cell into an embryonic cell into a heart, using studies where a certain thing, as hormone treatments and so on, cause developmental defects in mice and things like that, and we can use clues from that, and then a series of drugs to then change the media over. There's several different media changes with different drug exposures over a period of about 2 or 3 weeks, and then you get this beating sheet of cells. So that's the starting point.
So how do you use those then to test the toxicity or potential toxicity of a drug?
Well, okay, I think the step though that is missing, I think, with that's relevant to this story is that we started off with these beating sheets of cells, and what Kevin Healy asked, and we were collaborating with him, is that they asked, what if we now, instead of having the sheet of cells be covering the entire well, what if we put them into a very, very small container? So they printed or made little, very, very small holes in the tissue culture dish that were only 800 microns wide. So there's less than a millimeter in diameter. And they asked the question, what would confinement do to these cells? And I thought that they would be actually, all you'd get is now a very, very small sheet of cells as you use differentiation. But that's where the surprise came in this study. And the surprise came because the cells actually formed around the edge the fibroblasts grew and stuck it to the edge of this circle, this disc, and then instead of forming just what I thought would be maybe beating cells in the center of a target that was flat, the beating cells in the center actually ballooned up and then became sort of like a hot air balloon where it's fixed around the edges, and then it's ballooning up out of the dish so that it's actually taller than it is wide. And it's beating in the center with beating cells, and then it's hollow in the center and inside the balloon. And this is why we call it a microchamber, not just a sheet of cells.
So then how do you use it to test drugs?
So then this captures different aspects of development, obviously forming a chamber versus actually just a sheet of cells. And so what they decided to do is to test different drugs that were known to be toxic, cardiotoxic, and to see whether it could capture a toxicity that wouldn't be seen otherwise. And we used thalidomide, which is a drug which has been known to actually cause many types of developmental disorders, but the most serious ones were the heart defects. And sure enough, it actually caused a defect in these mini chambers, which was not appreciated when the same drug was given to mice. And unfortunately for the people who got thalidomide, it had been tested in mice and had been tested in animals and was deemed to be safe even in pregnancy because the mice didn't get any toxicity from it. But in fact, when we give it to these human mini chambers, cardiac chambers, they actually are really malformed, and so it would be obvious that there's a problem with thalidomide in this human model of development.
Would this only give us insight into the effect a drug might have on a developing fetus as opposed to an adult?
Well, I think that remember, the things that we're seeing are really small. You can see them, but they're very, very small. And so this is only modeling the very early stage of development. Now, one of the things we can do, though, is we can make the cells into different formats and even sort of stretch them and exercise them and things like that. And we can get them into a more adult phenotype. And a lot of what my lab actually works on and Kevin's lab works on is actually on drug toxicities in adult tissues as well. So there'd be different formats for trying out, really very, very early developmental stages versus modeling adult stages.
Well, your co-author, Kevin Healy, a few months ago unveiled a system of beating human heart cells on a chip that could be used to screen for drug toxicity. How does that differ from what was done here, and is that something that could address an adult population?
Well, it's hoped that it could, and I think that's the direction that we're going. And sort of tissues on chips we can actually potentially use for screening adult toxicities, and certainly that format and several other formats that we're working together on and so on is certainly something that we are studying. I think that the unique thing about this one is that that's only really primarily one type of tissue. We're just looking at the cardiac tissue. And if you look at the pictures of the microchamber, you actually see that the microchamber just has the cardiac cells are just one portion of what's developing. And then there's a whole other cell type, which we don't actually even know what it is, that is the layer below that, which is the majority of the cells. And then below that are these fibroblasts. So you have like a layer cake of different kinds of cells which are all conspiring together. And that kind of interaction between multiple cell types is something that is more difficult for us to model in, say, for instance, the tissue chips, that the other types of tissue chips where it's just cardiac myocytes.
Could either of these approaches, as it were, be industrialized? Can a ready supply be created economically for use in drug development?
Yeah, I think so. I think that's one of the big advantages of the tissue chip concept is that we can get the cells down to a small enough volume so that we can actually test potentially thousands or hundreds or thousands of different drugs with them. So I think that the reason to make things small is actually so that you can actually have you can test you can make many of them without having a huge cost of making the material. Actually, so making like a full, a piece of heart like the size of a bacon or something like that could cost thousands, tens of thousands of dollars, or perhaps $100,000 just to make that. And so you'd want to have something which is much smaller, so that it's economical enough so that you can test many drugs and test them over and over again to show that it's reproducible.
Well, you've done this work in heart cells. Is it transferable to other organ systems? Can we develop similar tests for, say, liver toxicity? Or
that's yeah. In fact, the liver is the furthest along. I think that there's actually already groups which have industrialized the use of liver cells on little microchips, and there are multiple other groups that are working on that. So I think that that's an area which is readily already developed. Neural cells could potentially be put onto chips as well. And I think that one of the wonderful, amazing things about the iPS cells and in particular is that they are poised to form any tissue in your body. So it's essentially just a trick. It's a matter of time before we can figure out how to make just about any tissue, and including these tissues which we've never had real access to. You for instance, I'm sure are very interested in medical research, but if I ask for a piece of your heart, just a little piece even, to study in our lab, and I told you that it would be cultured in a dish for a few weeks and then die, you probably wouldn't be too enthused about making that donation. But if I were to take some skin cells from you and make your cells into a limitless supply of pluripotent cells that could make unlimited numbers of cardiac cells, it would be a more interesting proposition, and obviously something that you can take further along, and that's just exactly the way things are. We could get heart tissue from a transplant situation, but it would only grow for a few weeks, and then you really couldn't reproduce it because you'd only have that limited supply of cells. And so it's really transformed the whole situation for the whole research setting, that we can get an unlimited supply of cells which we can actually engineer and do change their genomes and grow them essentially now really the limitation is really just the funding to get these cells made.
Bruce Conklin, a senior investigator at the Gladstone Institute of Cardiovascular Disease and a professor of medical genetics and cellular and molecular pharmacology at UC San Francisco. Bruce, thanks so much for your time today.
Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.